The overall goal of this experimental procedure is to induce and observe the cardiomyogenic induction of mesenchymal stem cells.
This method can help answer key questions in the mesenchymal stem cell field, such as whether MSCs can be induced by environmental signals to generate functional cardiomyocytes.
The main advantage of this technique is that it applies aggregate formation and cardiac feeder layers instead of pharmacological agents or genetic material.
The implications of this technique extend towards therapy of cardiovascular disease because it elaborates on the cardiomyogenic potential of mesenchymal stem cells.
Demonstrating this procedure with me will be Ms. Farwah Iqbal, a PhD student of our laboratory, Max Librach, and Nadav Gassner, both trainees of our laboratory.
After isolating rat pup hearts according to the text protocol, cut the ventriculi in half and transfer them to a 10 cm dish with 10 milliliters of PBS and pen strap on ice to let the blood wash out. Then, using curved scissors, cut the ventricular walls into small pieces with a diameter of 2 to 3 millimeters. Using a serological pipette, transfer the heart pieces from 10 to 12 animals to a 50 milliliter tube and let them settle. Remove as much PBS/PS as possible without removing any heart pieces. Then add 10 milliliters of fresh PBS/PS. To isolate the cardiomyocytes, after the heart pieces settle, use 0.15% trypsin in PBS to replace the PBS/PS, and incubate the tissue at 37 degrees Celsius with shaking for 10 minutes. Discard the supernatant.